So this is a chart showing the Stanley-Miller's experiment on the origin of life. So when you see the summary of main steps in the origin of life, so in the starting it was in the form of free atoms like hydrogen, carbon and nitrogen, they were free. Then inorganic compounds like hydrogen, water, methane and ammonia and carbon dioxide and hydrocyanic acids, they were present. Then simple organic compounds, molecules like methane, water, they form the sugars, fatty acids and glycerol, you can see here. And methane, hydrogen and ammonia, they are forming the amino acids. And methane, water, ammonia and HCN, they are forming the nitrogenous bases, purines and pyrimidines. So complex organic molecules were sugar plus sugar. So sugar and sugar, they joined together forming the polysaccharides. This one, monosaccharides forming the polysaccharides. Then fatty acids and glycerol, they are forming the fats. Amino acids and amino acids, they are forming the proteins. Nitrogen base, sugar plus phosphate, they are forming the nucleotides. And nucleotides, they are forming the nucleic acids. So in this way, the coacervates are formed here, which is aggregates of large complex organic molecules, and they can grow and they can divide. Then eobionts were formed, nucleic acid controlled coacervates, and free genes which can reproduce, self-reproduce, and these are the nucleoprotein combinations. Then first primitive cell, which was lipid protein membrane bound cell unit with the genes. So in this way you can see that how life originated from the simple free atoms, then inorganic compounds, simple organic molecules, complex organic molecules, coacervates, eobionts, free genes, and first primitive cell is formed. So Stanley-Miller experiment was there. It was just given. This theory experiment was proposed by the Stanley in 1953. So this is a chart showing the Stanley's experiment of life, how life originated in the starting. So experiment was carried out, and you can see here experiment. This was the Stanley-Miller. So name of experiment was spark discharge apparatus. It was used in the spark discharge apparatus. So what he did, you can see here that Miller's apparatus was called as spark discharge. This one, Stanley-Miller's experiment. Here you can see the spark was used here. Inorganic compounds were used, and then how the chemicals were formed here. Then later on Bahadur obtained all the possible amino acids, and Melvin and Kelvin produced amino acids and sugar by heating a mixture of hydrogen, water, vapors, ammonia and methane. So in the this experiment, Miller's experiment, this one, ammonia, methane, ammonia, water, 2:1:2:2 were sealed inside the spark chamber here. This one, CH4, NH3, H2O, and they were just sealed in the spark chamber. This is a spark chamber. Then another flask was used, which was half filled with the water. You can see here half filled water. Another flask is used here, half filled with water. Then H2O, water was heated and vapor released. So vapors were released from here, water was heated, condenser was used here. This is a cooling form. So water droplets are collected, then moving to the U-trap, and this is a boiling water, small flask burner. So vapors were released, then added to chemical mixture CH4, NH3 and H2, and H2O represent the water on the earth surface. So water vapors represents the water evaporating from the primitive. So in this way, it was artificially created the primitive atmosphere of early earth in the laboratory. You can see here how this produced from the inorganic materials. So they produced a few amino acids which were low level building blocks of life. So, but not all the amino acids were produced, nor sugars and purines, but both left and right and full forms, ended forms were later produced. No macromolecules were produced like proteins, DNA and RNA. So in this way, you can see this is a glass tube, this is electrode, electric spark is released there. This is a larger flask having the gases, then water vapors are produced under the condenser, under this pressure, and then they are able to collect the simple amino acids from them. So, and this is a condition under primitive atmospheric condition. This apparatus works. So, chemicals analyzed through the chromatographic and colorimetric methods. So, amino acids like glycine, alanine, aspartic acid, fatty acids like acetic acid, lactic acid, formic acid, and intermediate products like aldehyde and HCN were produced. So later on, HCN after irradiation, they produce the additional amino acids, purines and pyrimidines. So this was the experiment to prove that the life originates from the simple inorganic and then organic compounds. So this is a apparatus of Miller showing abiotic formation of organic compounds under primitive atmospheric condition. So this is all about the Miller's experiment, Stanley Miller's experiment, or you can say that formation of organic compounds under primitive atmospheric conditions.